Okay, so good evening, everybody. I'm delighted to welcome you to this year's annual address at the Institute of Neurology. I'm Mike Hanna, the director, and I'm very pleased to welcome Michael Arthur, the President and Provost of UCL, David Lomas, the Vice Provost Health, Alan Thompson, the Dean of the Faculty, and Pro-Vice Provost for London. I'm also really delighted to welcome our special guest lecturer this year, Dr. Richard Ewell. As we normally do, I'm going to try and give you an overview of the highlights of the past 12 months at the Institute over the next 20 minutes or so, and then I'll introduce our guest speaker. So a lot has happened over this last 12 months since the executive team met for our away day. And we couldn't have foreseen the dramatic events. Our vision remains the same, which is to be the leading translational neuroscience centre in the world and translate discovery science into experimental medicine, ultimately to find treatments for patients, alongside excellent student education embedded in research, and also work we do to try and overcome the fragmentation, which is a barrier to translation. We have a clear strategic operating plan, which we reviewed at our away day. This was devised following a Science Council review by Hugh Perry in conjunction with the faculty. And in the last year, we've particularly focused on supporting people, culture, equity, diversity, inclusion, and early career postdoc and early career support. But of course, everything changed this year with the COVID-19 pandemic. We had very interesting leadership nationally, which was somewhat confusing at times. Fortunately, we had very clear leadership at UCL, which cascaded down through the organization. And the entire community under this leadership has done, I think, an incredible job supporting each other, supporting our students, exhibiting kindness and contributing in a serious way to the national and international effort for COVID-19 in many different ways. I particularly want to thank my teams here at Queen Square, especially Helen Crutzen, the Institute Manager, and the support from the Deputy Directors, G.P. Schiavo and Linda Greensmith. And I also want to really thank all of the central ION professional services team and the departmental managers who worked extremely hard over this last 12 months during the COVID crisis. We also had fantastic support from the faculty office team, as we always do, but we had particular need for their support, obviously, over these last months. And it was also a great opportunity to further enhance my links and working closely with the leadership across the faculty, illustrated here. So, like many parts of UCL, Queen Square really swung into action and a lot of things happened. So over 25 of our PIs were directly involved in significant COVID research. We've already published 60 publications on COVID aspects. And our education team switched rapidly to an online blended teaching system for our 254 MSc students. There were also a range of important collaborations, both across UCL and also with the CRIC during this time. One example of an excellent piece of research done very rapidly and which received massive attention worldwide was led by Hadi Manji, Michael Zandi, Rachel Brown, and Ross Patterson. And they basically established an online COVID neurology MDT every week, where complex cases anywhere nationally at any neuroscience center could be discussed. And they were able to define the emerging spectrum of the COVID-19 neurology diseases, particularly in terms of the clinical features, the radiology, the laboratory findings. So this was really helpful nationally and internationally, and had a massive citation very quickly, as you can see here. We also developed the ways in which we translate our research into clinical benefit for patients. This is a good example of work between colleagues at the Institute and the Same You charity, where they developed an online neurological rehabilitation program, which was taken up, 2,000 sessions with patients happened in a short space of time. This was led by Alex Leff, working with Nick Ward and the team. We continue to be pretty productive on the publication front, with over 1,700 papers in this last year. That's about 5 every hour. And this included in what are considered to be leading journals. And of the 45 UCL academics cited in the global list of influential researchers, there were 11 from the Institute of Neurology listed here. We currently have £304 million in active research grants, and over recent years we've increased our income by about 20%, and we've increased our contribution by around 29% during this time. So I'm now going to attempt to highlight some of the research advances and the successes over this last year. Thank you to all the heads of departments who sent me literally hundreds of slides and papers, which I've tried to sift through and draw out a few examples. And I apologize to those of you where I haven't managed to include everything. So as part of our strategic plan, we did a reorganization of our departments a couple of years ago. Our 8 departments span basic science through translation to clinical research. And we're really interested in linking the basic science to benefits for patients. We have 5 women and 4 men who are fantastic heads of our 8 departments, illustrated here. And they've worked extremely hard, particularly over this last 12 months, to support all of their staff and students. I was particularly pleased that one of the new departments that we set up, led by Tony Shapiro and Tom Warner, has really been successful in securing major grants with over £15 million in just the last 12 months. And particularly notable were grants in relation to Parkinson's disease from the Michael J. Fox Foundation. And this department secured 2 major grants, as you can see here. The other one went to John Hardy, and actually there were only 4 grants of this size to the whole UK, so this was a massive achievement. The department have published in many important journals, many pieces of important work. One area of interest is, of course, the fact that glucose cerebrosidase mutations are a risk factor for Parkinson's disease, and this research suggested that ambroxol could be a potential therapy, and the efficacy trial, I think, is planned. The Wellcome Centre, led by Kathy Price, extended its Wellcome funding, which was great. They're now using the 7T scanner, which they installed last year, and are producing incredible images of the brain. And there were many successes indicated here. And I'd like to particularly congratulate Kathy and Tim Behrens for being elected to FRS. There was a 25th anniversary of the FIL, with previous directors assembling. This is now really the world's leading functional imaging laboratory. And in the lower panel, you'll see that Carl Friston was bamboozling Kirsty Walk on Newsnight about COVID modeling. Max Planck UCL Centre, led by Ray Dolan, continues to be highly successful, and their very successful international research school, after detailed external evaluation, was renewed from 22 to 28, which is a really great achievement. Neurodegenerative disease is led jointly by Sarah Tabrizi and Nick Fox. Nick Fox leads also the DRC component of that department. And again, they've been prolific in terms of their activities, major work in relation to cohorts that they follow. They've moved into clinical trials, particularly antisense, major publications, and John Rohrer now is one of the world leading experts in genetic frontotemporal dementia. The Huntington's Disease Centre continues to be highly successful. We all wait very keenly for the efficacy trial results from the Roche trial for the antisense approach in HD. Sarah, I'm delighted to say, was awarded the Alexander Morrison Medal at the Royal College of Physicians, Edinburgh, the Yara Award from the World Congress, and Sarah and Jill Bates, who head up the Huntington's Disease Centre, gave jointly the Jean Shanks Lecture at the Academy of Medical Sciences. And this department is spearheading our genetic therapy approach, which is really moving forward quickly at Queen Square. And we have several antisense, for example, now in human trials. And that's really just over the last few years, that's all happened very quickly, which is really great. Linda Greensmith heads up the neuromuscular disease department. There have been some important grant successes. The UCL Drug Discovery Institute sits in this department, and Paul Whiting renewed the centre for £12 million for 5 years. And Kenneth Harris achieved a £10 million grant from the Wellcome Trust to set up the International Brain Lab. The department undertakes research both from very basic molecular science, such as this RNA processing paper, right through to clinical trials in patients. Gene discovery is a big part of this department. Henry Holden leads for neurogenetics for Genomics England, and through his leadership, UCL and North Thames recruited more patients into the neurogenetics part of GEL than the whole of the rest of the country put together. Here's an example of a gene that they've recently discovered with Mary Riley, a loss of function mutation mechanism in the sorbitol dehydrogenase gene linked to fairly common forms of axonal neuropathy, which highlights an interesting link between diabetes and the mechanism of neuropathy with potential therapeutic implications. And they had the front cover of Nature Genetics. An important development in the past year has been the appointment of a clinical academic lead for ALS research, particularly clinical research at Queen Square, Andrea Malaspina, who starts shortly. This was a real joint effort between the hospital, the BRC, and the Institute to enable this position. And congratulations to Ricky Patani, who won the Graham Bull Prize and the Goulstonian Lectureship for his work on ALS. Dave Waring heads up the Brain Repair and Rehabilitation Department with a major focus on stroke. They've done excellent work in the genetics of intracranial aneurysms, along with other imaging research. And there have been many successes in this department. Xavier Golay has now got a UK patent on his devices to measure phantom devices within MRI scanners, called Gold Standard Phantoms. Alan Thompson was awarded the prestigious Sobek Foundation Prize for MS research, and Valerie Iodici secured £1 million for a randomized controlled trial in autonomic failure. Karen Duff is the head of the DRI department at ION. This is, of course, the UCL centre of the 6 centres that form the DRI. And of course, UCL is the headquarters of this national effort led by Bart de Strooper and Adrian Ivinson. I just heard last night, I think it's just been announced today actually, that Karen has been awarded the Outstanding Contribution to Neuroscience Award by the BNA, which is a tremendous achievement. Karen hasn't been here that long, but her group and the groups in the DRI are already producing really high quality mechanistic research to help us understand dementia. And members in the DRI made major contributions to a special section of science recently on neurodegeneration. In epilepsy, we have a huge successful team linking basic science to clinical research. Just to highlight John Duncan's work, developing a navigation tool to enable the precise placing of intracranial probes to record electrical activity prior to planned focal resections in focal epilepsy. And we're all very excited as the gene therapy for refractory focal epilepsy programs advance. So the first sort of compound, if you like, which is the lentivirally transferred potassium channel to overexpress the potassium channel, is now close to moving into patients with clinical trials, hopefully at the beginning of 2022, led by Dimitri Kuhlmann and his team, but a whole sequence of other potential agents in the pipeline is very exciting. Neuroinflammation is led by Olga Ciccarelli. She was recently appointed to head in this last year, and also has been appointed as deputy editor of the Journal of Neurology. So congratulations to Olga. The MS group and the Department of Neuroinflammation continue with their highly successful imaging trials and basic science programs. Examples illustrated here. The trials programs led by Jeremy Chataway. So we have this fantastic Leonard Wolfson Experimental Neurology Center at Queen Square, led by Professor Vincenzo Libri, and a whole raft of first-in-man industry-sponsored trials are now happening. We now have 13 antisense oligonucleotide studies in neurodegenerative diseases, including these diseases illustrated here, which is quite amazing. They've done 1,000 lumbar punctures and 200 intrathecal injections of antisense agents. And they also contributed as a center in the Oxford COVID vaccine trial. One of our strategic objectives over the last few years has been to increase our enterprise activities, and this has been led by Paul Whiting. And I'm really pleased to see so many PIs who previously were not linking into form partnerships with companies are now doing so. For example, Paola Giunti secured an impressive grant to look at antisense in ataxias. We can't do any of this without key partnerships. I'm particularly grateful to Alan Thompson and David Lomas and their support and guidance and leadership from the faculty in the school. We have a great relationship with the hospital team, Chris Turner and John Melbourne, and we've worked really hard to develop things, for example, the ALS professor I mentioned with the BRC, and I'm really grateful to Nick Wood. But these partnerships are really important. I was sad to see that Professor Marcel Levy is leaving. He's been the chief executive for the last few years. He's been an inspirational leader, leading by example, and has really pioneered the research hospital concept, which has been really great for the Institute in particular, but UCL more widely. Key partnerships in terms of helping us raise funds include the Office of the Vice Provost Development, and I want to thank Laurie Houlihan for her inspirational leadership in recent years, and also thank Adrian Punax. We also have great partnerships with the Brain Research UK team and the National Brain Appeal and other key patient and charity organisations. So we're really committed to our EDI mission, that's equity, diversity and inclusion at the Institute, and we're really working hard to harness the power of diversity to develop and deliver our mission. I was really delighted that we managed to secure renewal of our Athena Swan Silver. I want to thank the entire SAT team and the leadership of Lizzie Fisher and Nick Wood. But I'd also like to thank all the colleagues who have made this a continual chain of Athena Swan success for the last 10 years. We got bronze led by Mary Riley. We got the first silver led by Alex Leff. There was then really important work by Selina Ray and Helen Plum-Favreux before we got the silver renewed this time. So thank you to all of those colleagues and all the SAT teams. We've got new project manager to boost our efforts in Athena Swan, and we're really pleased to have Anna de Souza Ifaro on board. And we've now appointed a new Deputy Institute Director for Equality, Diversion and Inclusion, Helen Plum-Favreux. And they've created a really nice website that you can have a look at if you want to. We had great success in senior promotions. I've got 2 slides full of the successful candidates, and importantly, the balance is 8 women and 9 men, which is great and a big change from when I first started. These are the successful candidates illustrated here. I won't read all the names, but congratulations to all of these colleagues. It's a really huge achievement to get promotion at UCL. So well done to all of them. Also, very importantly, we've been successful in supporting early career researchers, and here are some success promotions in the last 12 months. So well done to all of those colleagues. And it's really important that we support our very best people for their futures. And here are 4 proleptic appointments. These are colleagues who've secured 2 externally funded senior fellowships before achieving proleptic status. So congratulations to all of them. 2 of our colleagues, Mary Riley and Bart de Strupe, were elected to the Fellowship of the Academy of Medical Sciences. We now have 29 fellows at the Institute. I was pleased to be elected to the council, and I'm hoping that David Lomas, who is also on the council, will show me the ropes. We had 1 retirement this year, John Rothwell, who did fantastic work over the years in neurophysiology and was a great colleague at the Institute. Sadly, there were 2 deaths, Lindsay Simon, previously consultant neurosurgeon at Queen Square, and Robert Balaz, who was head of the MRC Departmental Neurobiology Unit. So I can't do an annual address without telling you what's happening with the building. So in 2011, this was our line diagram of a building in the goods yard, kind of connected to Queen Square House, but we hadn't identified any funds. Fast forward to 2020, and thanks to huge work by many people, including the Provost, David Lomas, Alan Thompson, and many others, we have approval from council now to progress to build this fantastic new building on Gray's Inn Road, which will be ready in 2024. When it was approved by council, there was huge interest on Twitter with over 23,000 impressions in 24 hours just for these 3 tweets alone. The building work has started, or I should say the demolition has started. Here are the images of the demolition. And thanks to Kevin Argent and his team and the UCL Estates team, and also the sponsor, Alan Thompson. This is what it's going to look like, which is absolutely fantastic. And crucially, I think there's going to be co-location of patients and science in the same building. The partners in this building are, of course, the UCLH patient services, particularly outpatients and MRI, our scientists, particularly wet lab science, and of course the DRI headquarters. A crucial part of the new building, which is quite different to how we work now, are going to be a whole set of new core facilities, and this has been led by GP Schiavo with new technologies, new ways of working, including human imaging, super resolution microscopy, and a whole bunch of automated processes that we do not have now in any sort of coordinated way. And we believe that this new way of working will connect patients and their samples with research, because a lot of these systems are to analyze patient samples. They'll connect scientists to this new technology, and also scientists to scientists in a different way, which is exciting. Of course, we'll retain buildings around Queen Square and 3 floors of Queen Square House. And we envisage this dual hub system in which we have experimental medicine research more at the Queen Square end of the hub and more basic science at the Gray's Inn Road end. But importantly, there'll be the mixing of patients and scientists at both places, which I think is really important in a translational neuroscience community. So this is a fantastic opportunity, but there are important challenges. It's ensure the critical relationship between the hospital and institute continues to be successful and grow, because I think this underpins our translational success. We do need to work to get full buy-in of all colleagues about this new way of working with core facilities. We are continuing our efforts to raise 27 million towards the core facilities, and clearly it's really important the ION community remains cohesive as they are now over the next 4 years to really maximize this opportunity. There's a great website that Anna Phillips developed about the new building, which you can visit. Turning to education, this is a crucial part of what the Institute does. We've had a very large increase in students over the last number of years, 300% increase in MSc students, 160% increase in PhD students, with over 500 in total now. And I want to thank David Blundred, Alex Leff, Steve Kennelly, and Dimitri Corbin for their great work supporting our student education activities, with also great support from Julia Evans and David Shanks in the faculty. We were very pleased to get good scores in our postgraduate taught experience survey. These are the progressive improvements over recent years, and this contributed to a great performance by the faculty overall. I particularly want to congratulate David, who has been a fantastic education manager and won a UCL Personal Excellence Award, which is very well deserved. I also want to just highlight that our MSc in Clinical Neurology by distance learning is equal top of 1,695 courses across UCL in terms of diversity. And they've done a great job in driving diversity on this course, and we can learn a lot from this work. We're expanding our PhD programs. We have 264 students at the moment, but Pat Harris, who was a great servant of the Institute for 25 years, sadly died recently, and her legacy, she left funding which has allowed us to establish the Pat Harris Neurodegeneration Disease Fellowship, a 4-year basic science PhD. We've also entered into an exciting new agreement with Cleveland Clinic London for 6 new neurology or neurosurgery PhD fellows to start next year. So thank you to all the fantastic education team. Finally, the student prizes. So this year, the Queen Square Essay Prize, we couldn't separate the 2 candidates here, so it's been given jointly. So congratulations to Edwin Jabari and to Paul Zun. The Heyman Gorloff Prize goes to the MSc student in clinical neuroscience with the best overall mark, and that goes to Sophie Newton, so well done. And the Hugo James Ross Prize is for the highest mark in the MSc in clinical neurology, and I'm pleased that goes to Huda Aldin. The graduation this year was a bit different, but I was really proud to read out the names of the 81 graduates from the Institute last week, and it was great to see 71 Institute of Neurology staff in attendance at this graduation. I know that the Dean was really pleased. He tweeted at the end of the week because he'd overseen the graduation of 1,400 students, and he looks pretty pleased and proud of all those that have graduated. So we can't actually unmute you to clap, but I do feel that we should applaud in some way the great achievement of the students that have won the prizes and the students that have all graduated. So we have an artificially engineered applause, which Derek is going to activate, and which I hope you'll feel. I don't think we heard that, Derek. You need to do it again. Great, well done, Derek. Okay, thank you. So finally, I just want to sincerely thank Michael Arthur for his fantastic help to the Institute of Neurology. I know all colleagues of the Institute are very grateful. I know he spent probably a lot more time on the Institute than perhaps he should have. I expect he may never forget a building called Queen Square House or the Goods Yard, but you never know. But of course, more widely, he's been a fantastic leader for UCL and has been critical in his leadership, in meaning that UCL is now firmly within the top 10 universities in the world, which is amazing. So we all just want to sincerely thank you, Michael, for all of your efforts. We thought we might play that applause again, but then we thought it wasn't serious enough, so we decided not to, but just to thank you sincerely for everything you've done. We really appreciate it. So I'm now going to move on to introduce our great guest speaker. So Dr. Richard Ewell received his PhD from the University of South Carolina, where he worked on the mechanism of action of the protein toxin ricin. He then joined the laboratory of David Neville at the NIH for postdoctoral work focused on engineering protein toxins for biomedical applications, a theme that he further expanded at NINDS as an independent investigator working on immunotoxins as new strategies for brain tumors. His laboratory elucidated the molecular mechanisms of programmed cell death, showing how BCL2 family members control cell survival. His long-term interest on how mitochondria control cell homeostasis and cell health drove his most recent discoveries on the molecular mechanisms of mitophagy and mitochondrial quality control and their causal links with neurodegenerative disorders, in particular Parkinson's disease. For these recent discoveries, Dr. Ewell has been awarded the 2021 Breakthrough Prize in Life Sciences, which is a massive achievement, as you all know. In addition, he's been awarded 3 NIH Directors Awards. Dr. Ewell has published more than 180 peer-reviewed articles and 16 patents, and has widely contributed to the formation and training of the next generation of scientists, including students on the NIH UCL PhD program. So it's a real honor and pleasure to ask Richard Ewell to give the 2020 annual address. We're very much hoping that his link is going to survive. We've had a few technical problems. We do have a recorded version of the presentation, so if we're having difficulty, we will revert to that, but we hope that won't be the case. So I'm going to ask Richard to share his screen and hopefully commence his presentation.
Thank you, Mike, for that kind introduction, and thank you to the UCL community for the honor of giving this annual lecture. It's also a great pleasure because of the many close colleagues I have at UCL. So, as Mike mentioned, I'm going to talk about the intertwined roles of 2 Parkinson's disease genes and how they're linked to mitochondria and how they're linked to innate inflammation and Parkinson's disease. Now, as you all well know, the severity of Parkinson's disease and that it's late onset, largely idiopathic. And without knowing the cause of the disease, it's impaired developing therapies that change the long-term demise of the patients. But over the last 10, 15 years, a number of monogenic disease genes have been identified, and understanding what they do not only may help those patients, but may also help understand the etiology of idiopathic Parkinson's. Now, we know LRRK2 and alpha-synuclein, genetically linked to Parkinson's disease by Andy Singleton and John Hardy. They are dominant mutations, likely gain of some toxic function, and it's still not clear what that toxic function is. PINK1 and Parkin, they're autosomal recessive, indicating their loss of function genes. So it would be very important to know what their normal function is. I mean, clearly in man, a normal function of them is to prevent Parkinson's disease, but what do they do at the molecular or cell biology level? So Parkin was linked to Parkinson's disease by Hattori and his colleagues in Japan. And just looking at the sequence of the protein expressed from that gene, one sees a ubiquitin-like domain. So that's homologous to the post-translational modifying ubiquitin, which is 70 amino acids long. And then it's got these domains that are predicted to be E3 ubiquitin ligase. So it's kind of interesting, a ubiquitin ligase and a ubiquitin-like domain on the same protein, and indeed has been shown to be an E3 ubiquitin ligase. And it's located in the cytosol. Now, another monogenic cause of Parkinson's discovered here at UCL and Nick Wood is PINK1. And when his group looked at the sequence, they could see a predicted kinase domain, and it is a kinase, a membrane spanning domain, and a mitochondrial targeting sequence, a algorithmically predicted mitochondrial targeting sequence. And his group then indeed showed it was targeted to the mitochondria. So like the other Parkinson's disease linked genes, there's no reason to think a cytosolic protein and a mitochondrial protein would be working in the same pathway. But they could, of course, but there's no indication of that. And immediately after the discovery of these genes, people around the world made knockout mice, knocking out Parkin or PINK1. They've even made double and knockout mice. And unfortunately, these mice do not have any substantial phenotype, and particularly no phenotype linked to dopaminergic neuron loss. And that stifled the field for a number of years, in my mind, until a fly geneticist, Leo Planck at the University of Washington, he identified Parkin mutant flies. And these Drosophila had a phenotype. They had a very severe phenotype. They lost some dopaminergic neurons. They had a muscle loss, and they were male infertile. And then shortly after, Ming Guo at UCLA and Kai Chuang in Korea identified pink one knockout Drosophila, and they phenocopied the Parkin knockout flies. And when you cross them together, it's no worse. And other epistasis experiments indicated they worked in the same pathway. So how would these two proteins work together, and in what process? So this is a summary of the first part of my talk. I'm going to outline a pathway where they work together, and I'm going to highlight a couple steps and show you the data from a couple steps that were important in this pathway and pioneering molecular pathways in all of biology. And the idea, the model is that cells can have hundreds and hundreds of mitochondria, and if one gets damaged, this kinase, pink one, accumulates on the outer membrane. And from there, using its kinase function, it will recruit this Parkin protein from the cytosol to the mitochondria. And that turns the E3 ligase activity on. It ubiquitinates the mitochondria. It eliminates them by autophagy. And therefore, we suggested it comprises a mitochondrial quality control pathway, because only upon mitochondrial damage do they get eliminated. And this builds on pioneering work from Tony Shapira here, already years in the making, linking mitochondrial damage to Parkinson's disease. So our first experiment that led us in this direction was by a graduate student of mine, Derek Narendra. He went on to finish his PhD with John Walker at Cambridge in one of these joint programs that I've now got with UCL. And what Derek did is he fused Parkin to GFP and expressed it in cells. These are HeLa cells. It could be many cell types have now been studied, including neurons. Mitochondria are labeled in red. And indeed, just like the literature shows, Parkin's clearly cytosolic, and pink one would be localized to these red mitochondria. So then Derek decided to stress the mitochondria in different ways, and he added a fairly mild stress, which is a small molecule that will depolarize the proton gradient. And these are not that stressful because they can be washed out and the mitochondria resume their normal function. So when you add one of these compounds, in this case, it's called CCCP, it's a depolarizing agent. I'll show you what happens to the Parkin. This is accelerated, it's about 30 minutes in real time. So we add the CCCP and watch what happens to the green Parkin. It very robustly migrates to the mitochondria. And this process, by a number of our experiments and other labs, is 100% dependent on pink one. So pink one is in some way, in some conditional way, upon mitochondrial depolarization, recruiting Parkin. Now you don't need to add chemical treatments to see this. This happens normally upon excessive exercise or damage to mitochondria, or in genetic lesions. Now, to test the idea of depolarizing a subset of mitochondria, we turn to the MFN1, MFN2 knockout cells. These are genes, proteins involved in mitochondrial fusion. And David Chan at Caltech generously gave us these cells, and he had already shown that if you knock out one of the mitofusins, the mitochondria appear quite normal. And if you knock out another one, they're quite normal. But when you knock both of them out, about 10% of the mitochondria lose their membrane potential. They're impaired, they're not functional, and they lose membrane potential. So we thought this would be an interesting place to examine Parkin localization, looking at this genetic lesion. And when Derek expressed GFP Parkin in wild-type mouse embryonic fibroblasts, you see it's cytosolic in, let's say, 98% of the time it looks like this. Same with the MFN1 and MFN2 knockout mouse. But when you express GFP Parkin in these double mutants, about 90% of the time, the Parkin is localized in these small spots. And if we blow up on that image of those cells, So we take this domain here of a MFN1, MFN2 double knockout. So it's expressing parkin. We can see parkin is localized in these spots that we've just seen. And then if we stain for cytochrome C to look at the total mitochondria, we see there's a lot more mitochondria than there are parkin spots. But the parkin does localize to certain mitochondria, but not all of them. So then we look at mitotracker, which is a measure of membrane potential. And you can see, for example, these two mitochondria, the right one maintains membrane potential, the left one does not. And if you look at the parkin, it's on the left one. So if one mitochondria loses membrane potential right next to a healthy one, the parkin is recruited there. And this also depends on PINK1. So somehow PINK1 is sensing the state of the mitochondria and recruiting parkin upon loss of membrane potential. And so how does this PINK1, I mentioned it accumulates, how does that work? How is this mitochondrial damage sensed? So when my postdocs started working on this, they did a Western blot against PINK1, and you don't see much. And the literature had already noted that, and people thought the antibodies don't work. But remember, we found if we add CCCP that causes parkin recruitment in that movie I showed you, when we add CCCP, we see a very robust band at the exact molecular weight of PINK1 using the same antibodies. But since the thoughts were that these antibodies didn't work, we had to go to the nth degree and prove, and this is indeed PINK1. And the really important advance here was when we found that if we wash out the CCCP, remember, these are readily reversible, we wash out the CCCP for 2.5 or 5 minutes, and the PINK1 immediately goes away. So that told us that it's being turned over very rapidly. It's being produced and degraded normally, and the CCCP stabilize it. So, and then it's being degraded again. So we screened all the proteases in the mitochondria using RNAi, and we identified PARL as a rhomboid protease in the inner membrane. So to confirm this, we got knockout MEFs from Bart de Strooper and confirmed that this was the protease mediating PINK1 destruction in lane 1. So you look at lane 1 here, it's not eliminated. And when you add CCP, there's a slight change due to the mitochondrial targeting sequence, and you wash it out. So we identified PARL. We also found if you run, this is a SDS gel, if you run a blue native gel to look for high molecular weight complexes, we found that PINK1 almost completely runs at a very high molecular weight band that we identified as the TOM complex. This is the complex of protein import into mitochondria. So these papers, this work, along with several other labs, lead to this model of the PINK1 life cycle. Now, in healthy mitochondria, it's got a mitochondrial targeting sequence. It's being imported through the TOM complex into the TIM complex. It's being cleaved by PARL, and this generates an N-terminal phenylalanine. And this was discovered by the Nick Wood group in this paper right here. So this is an important clue into the life cycle. And also in this paper here, we found that the importance of TOM7, I'll get to in the next slide. And so we'd already known from prior researchers that the proteasomes involved in eliminating PINK1. We knew PARL cleaves it here, but this was a bit of a mystery until I had a very clever postdoc, Koji Yamano, and he said, well, he just knew from studying that phenylalanine is an N-degron from the N-end rule pathway discovered by Alex Vershovsky. And indeed, he subsequently clearly demonstrated that this phenylalanine triggers ubiquitination by these N-end rule E3 ligases and proteasomal degradation. So the net sum of this in healthy mitochondria is to generate this. It's being translated, imported, clipped by PARL, degraded by the proteasome, and doesn't exist. Now we know PARL's localized specifically on this membrane, and we know that membrane potential is required for import into the TIM complex. So if we add CCCP, it prevents this import. So that prevents PINK1 from ever seeing the PARL. It's not clipped, and it accumulates on the outer membrane, bound to the TOM complex via this specific subunit, TOM7. And from there, with its kinase domain, it somehow recruits parkin from the cytosol to the mitochondria. So how does PINK1 recruit parkin? So people thought of we spent 5 years working on this. And actually some people thought, well, maybe PINK1 phosphorylates parkin. That'd be kind of the simple solution. And several papers appeared, we couldn't confirm them until this paper came out from the Mitochondrial lab. And I hope many of you remember him because he got his PhD at UCL in the Nick Wood group. And his paper says PINK1 phosphorylates parkin at serine 65 in the ubiquitin-like domain. This was the first paper of a series that we easily confirmed. It's absolutely clear and true that PINK1 does phosphorylate this serine. We mutated this serine to an alanine so it couldn't be phosphorylated. And we found that PINK1 could still recruit parkin to the mitochondria via its kinase function. So we kept seeking the substrate. And remember, this is a ubiquitin-like domain and it's phosphorylated by PINK1 at serine 65. And it was just, I won't go through all the technical steps, but my postdoc came in with this sequence one day and it was just really so gratifying and clear. She had identified that PINK1 phosphorylates ubiquitin. Now, a ubiquitin kinase had never been discovered before, but the result that was so compelling is her identification of serine 65 in ubiquitin as the substrate site. That's exactly homologous to the site Mitochondrial had already shown PINK1 phosphorylates. So it was easy to confirm by many different ways. And then the second discovery in this same project was looking at, so what could phosphoubiquitin do? Well, we already knew that parkin was an E3 ligase, but it's actually a very poor one. And if you look at its ladder of ubiquitin, this is a measure of ubiquitin conjugation. If you just incubate it with ubiquitin and all the enzymes involved in ubiquitination, you don't see much activity about, or if you expand the sensitivity, you might be able to see something. But what was really exciting was when you add in phosphoubiquitin, if you add it into the mix, it turns parkin from the off state to the on state. So here's an E3 ligase being activated by a phosphorylated ubiquitin produced by PINK1. And to summarize, this is our paper on it, but the Mitochondrial lab published independently, discovered the same thing, and so did the Matsuda lab in Japan. And we all published in the same month. And a whole series of subsequent papers have expanded on this, this discovery. And so the way PINK1 recruits parkin is it ubiquitinates, PINK1 phosphorylates ubiquitin that's already present on the mitochondria. And that recruits parkin, binds to that phosphoubiquitin, and then PINK1 also phosphorylates parkin, and that turns it to on state, and it ubiquitinates mitochondria that lead to mitophagy. But also, I just want to point out that, like in my title, the intimate link between these two independently discovered Parkinson's disease genes. So if there's ubiquitin always present on a steady state in outer membrane proteins. When mitochondria get damaged, PINK1 gets stabilized. Then PINK1 phosphorylates these chains, and these act as receptors so that inactive parkins recruited from the cytosol bound to phosphoubiquitin on the surface, and that activates parkin. And parkin then ubiquitinates more proteins on the outer mitochondrial membrane. And so the product of parkin then becomes the substrate of PINK1, and the product of PINK1 is the activator of parkin. So this is the first identified kinase for ubiquitin. It's the first identified function for phosphoubiquitin, and it shows that PINK1 is a ubiquitin kinase and parkin is a ubiquitin ligase. So I just think it's just fascinating to understand as we go forward how these two evolved to be so intimately linked together. And this pathway that I've covered has been corroborated and expanded even at the structural level. There's the first full-length structure of parkin from the Kaylee Gehring lab. And you can this is the cytosolic form, and if you know the function of E3 ligases, you can predict this is inactive. And parkin is inactive in the cytosol. And this is the structure of PINK1. This is determined by the David Commander lab at the LMB in Cambridge. And this is PINK1, and this is ubiquitin fitting right into the active site. And you can see serine 65 fitting right into the kinase active site. And then Commander determined the structure of parkin bound to the product of PINK1. And you can see it fitting in here, causing a dramatic conformational change that predicted and actually turns parkin into the on state. So we've now unraveled this new mechanism of mitochondrial recognition of damage by PINK1 and how PINK1 recruits parkin and turns it on. And how about these ubiquitin chains? I mentioned they cause mitophagy, but I haven't shown you any of those results. So if we look at stained cells, a field of like 20 cells, and DAPI stains the nuclei, and the green stain is for mitochondrial DNA. And you can see there's lots of little green dots. That's the mitochondrial DNA. And if you depolarize with like CCCP for 3 hours to activate the PINK1 parkin pathway, you see the mitochondria quite dramatically clump together. And by 24 hours, overnight of depolarization, you see the mitochondria are completely absent in most of the cells. I have Derek Narendra, the grad student, first discovered this, and we couldn't figure out, we'd never seen anything like it. No one else had it either. And not only the mitochondrial DNA, the entire mitochondria are gone. So we thought that's how we got into the field of autophagy. How could one eliminate all the mitochondria? So we looked to test whether it was involving autophagy. We looked at in a cell line that had a loss of an essential autophagy gene, ATG5. And we repeated that same experiment. And you see these mitochondria, after depolarization, they clump up, just like in the wild type. But after 24 hours, they do not disappear. They stay clumped up, indicating that autophagy is what's eliminating the mitochondria. And that's why we call it mitophagy, for autophagy of mitochondria. Now, how would ubiquitin chains be involved in this? There had been a dogma in the field that adapter proteins like P62 that are known to bind ubiquitin and bind LC3 could recruit cargo like mitochondria into an autophagosome, and that would be the recruitment mechanism. So we knocked out, actually Komatsu in Japan made a P62 knockout mouse, and he was very generous. He was doing the same experiment, but regardless, he sent his knockout MEFs of P62 knockout cells, and we saw no defect in PINK1 parkin mediated mitophagy. He was doing the same work, and we published back to back. So then what is causing it if it's not P62? Well, we didn't know, but there's a whole series of autophagy receptors, NBR1, NDP52, TAX1BP1. So we didn't know if any of these were involved or some redundant combination. So we started out by just knocking out all 5 of them with CRISPR. So we knocked out all 5 of the autophagy receptors to see if it had any effect. And if it did, we could hone in on which ones. But if it didn't, we could think of something else. So if we look at those knockout cells, I just inserted those in here in this last slide. So here's where we've eliminated them all normally. And in the 5 knockout, you see 2 changes. First of all, you see the green mitochondria do not clump up, and you see they're not eliminated. And it's a complete block in mitophagy. So we knew at least one of them was involved. So we did rescue experiments. We took the 5 knockout line, and we put each of these individual ones back in. Here shows the rescue. And we confirmed high level on the graph means no mitophagy and low means lots of mitophagy. We confirmed that P62 has insignificant activity, same with MBR1, a little bit with TAX. NDP52 was the most active, and since it was expressed less than the optineurin, we rank order these as NDP52, optineurin, TAX. And this is, I bring it up because it's particularly interesting because these two autophagy had previously been linked to the autophagy of bacteria. So that's called xenophagy, and it's a host defense pathway to eliminate invading bacteria. So the same 2 involved in, this is the Felix Randau group in LMB and the Ivan Dikic lab in Germany, reported these results. And so it's really interesting that there's a link between mitophagy and xenophagy at this level because, mitochondria, once upon a time were bacteria. And also it's interesting that I didn't get into it, but there's a kinase, TBK1 involved in this process, and both optineurin and TBK1 are risk alleles for ALS. So I've kind of moved you through the highlights of this pathway. And now I want to switch gears and talk about what's the role of this in vivo and what is the pathophysiologic relevance to Parkinson's disease. So there's quite a few papers now by different groups. This is actually not our work. We published this 2 years ago, and this is work from another lab that confirms and extends it. But this is a measurement of mitophagy in heart tissue. There's mitochondria that's in the matrix that turns from green to yellow. You can do some optical scanning to determine the mitochondria are in the lysosomes. And what we found is if we exercise the mice by treadmill running, exhaustive exercise, and this group did as well, there's more than a doubling of the rate of mitophagy. And the important point I want to get to is if you do the same thing with PINK1 knockout mice, there is no increase in mitophagy upon exercise, pointing to the in vivo function of PINK1 in this process. And I just want to point out, this is from this other group's paper. There's another probe being used to study mitophagy that's localized to the outer mitochondrial membrane. And like a recent Cell paper from Japan, who also pointed this out, this mouse has a high background rate of fluorescence, and it seems to be obscuring this effect that one sees with the matrix probe, and that we see, this other group sees no difference in mitophagy. So it can occur in vivo, but what's the implication with Parkinson's disease? So we know for many years that inflammation is linked to Parkinson's disease. Lots of cytokines are found to increase in the blood, particularly IL-6 in many studies, but it's not been clear whether it's cause or effect. I mean, it wouldn't be a surprise to have inflammation causing neurodegeneration, but it also could be that loss of neurons may cause the inflammation. And then we got particularly intrigued when a series of papers came out showing that mitochondrial DNA can trigger inflammation. For example, after heart failure, mitochondrial D that escapes from autophagy can increase inflammation. There's links to the different inflammasome components. And this important paper by Jerry Shadle showed that the mitochondrial DNA activates the CGAS sting pathway. This is a pathway of sensing cytosolic DNA. And we recently reported that PINK1 Parkin, the absence of them, that they normally prevent a sting-induced inflammation in mice. But this is a bit difficult because I mentioned PINK1 Parkin knockout mice don't have any Parkinson's disease relevant phenotype. So here we had to stress the mice in various ways to induce a phenotype. So I just want to show you some new unpublished data that are more clear and more definitive. I'm going back to this work I mentioned earlier. Leo Palank discovered Parkin mutant flies. And these are wild type flies, and this is how they hold their wings. But Parkin mutants, they have a collapse of the thorax, which is this big muscle on their back that's involved in flying. And when it collapses, the wings flop out like that, or they flop down by their side. So here we have a phenotype that occurs innately. We don't have to stress it. It's like humans. They innately get Parkinson's disease when they're missing this gene, and the flies innately show this wing phenotype. And there's been links to PINK1 and Parkin in Drosophila to innate immunity. There's this paper from the Planck group talking about innate immune responses in Parkin mutant flies, and Miguel Martins, you may know, he's up in Leicester. He's shown similar stuff with PINK1 mutant Drosophila have increased innate immunity. So what we did is we crossed Drosophila into two different loss of sting alleles. Here's a wild type fly and a wild type sting. And you see, this is a normal thorax. This is under a microscope. And these are little bristles. They're all lined up showing that round curvature. And this is in a wild type sting. And if you have wild type sting in a Parkin mutant that Leo Palank discovered, you see a big concavitation of that thorax. Now, when you take either one of these independently derived sting alleles and you cross it into a Parkin mutant, it rescues that phenotype. And it's quite robust. So wild type mice have very little indentation. The Parkin mutant flies have, in our hands in this experiment, 76%. And if we cross them into this loss of sting allele, that 76 goes down to either 6% or 14%. So a very substantial rescue of this phenotype, indicating this phenotype is being caused by sting. Sting is somehow mediating this process. So there are also, these flies can't climb the vial very well. So I'll just show you the left vial are wild type flies. This vial is Parkin mutant flies. They can't climb well. And this vial is Parkin mutant crossed with a sting mutant. And you'll see this guy, Andy Molman, will shake them down to the bottom and we'll watch them climb up the side of the tubes. See the wild type climb right up? These are climbing right up, but these are in pairs. We not only rescue the appearance, we rescue their activity. And surprisingly, I mentioned male Parkin knockouts are male infertile, but when we cross them into a sting null, we rescue the fertility. For example, 156 adults came out of these crosses in wild type, none in the Parkin knockout because they're male fertile. The sting seemed to be slightly less fertile, but when you have the double cross, you regain the fertility. So this is opens the door for a lot of new studies. And I just want to mention Alex Whitworth at the Mitochondrial Biology Unit in Cambridge. He recently reported different results from this, and he's been very generous to send us his flies. So we're going to sort it out, and we've now confirmed this with 2 different sting alleles and 2 different Parkin alleles. Now that brings me back to another species that has a phenotype with loss of pink-1 Parkin, and that is humans. I mean, and so we contacted Christine Klein, who has a large cohort of pink-1 Parkin patients in Lübeck, Germany, and Max Bors is first author on this study. He's a resident, and they looked at IL-6 levels in the blood of biallelic pink-1 Parkin mutations. And you can see a highly significant increase in this is a mixture of the Parkin and the pink-1, both biallelics with PD compared to healthy controls. Same with an Italian cohort, a very significant increase in circulating IL-6. And in this cohort, they also compared it with idiopathic and found an increase, significant increase. I mean, as I mentioned, it's already known that IL-6 is increased in idiopathic PD. They found a significant increase in the pink-1 Parkin mutants relative to the idiopathic. And they also looked at another inflammation marker, C-reactive protein, and found a highly significant increase in circulating inflammation relative to idiopathic Parkinson's, and a good correlation between the CRP protein and the IL-6 in the patients. And this is the most exciting result they got for me of all, is they looked at circulating mitochondrial DNA levels in the blood of these patients. As I mentioned, prior studies with heart attacks and things, circulating mitochondrial DNA is poor prognosis. And what they found is relative to either healthy controls or idiopathic, there was a increase levels of mitochondrial DNA in the blood of these patients. So I think this is completely consistent with a model of mitophagy, preventing, eliminating damaged mitochondria and damaged mitochondrial DNA to prevent inflammation. So just to summarize this part, I've got a little bit of new results to mention, but we suggest that loss of pink-1 Parkin sensitizes flies, mice, and men to inflammation. We think it's caused by the insufficient disposal of mitochondrial DNA that can activate inflammation via the cGAS sting pathway. I'm going to get into more of this in the next slide. And if you look at where innate immune pathways arose during evolution, and if you look at where Pink-1 arose during evolution, they're concordant. So one can think that maybe before innate immune pathways, since mitochondria look like bacteria in certain antigenic ways, it didn't matter so much if your mitochondria get damaged. But when you developed potent innate immune sensors that sense bacteria that would cross-react with the mitochondria, it can be important to rapidly and efficiently eliminate these damaged mitochondria. So now I want to talk about a little bit of exciting new work, I think, on the cGAS STING pathway. So this pathway, the STING pathway, had long been known that to activate interferon production as an innate immune process. And more recently, the cGAS was discovered by James Chen, and that is a sensor of cytosolic DNA, either viral DNA, bacterial DNA, or as I mentioned, several papers show mitochondrial DNA can, if it's released in the cytosol, can activate this cGAS to produce a dinucleotide that activates STING, and then STING moves from the ER to the Golgi, and via this TBK1 that I already mentioned in ALS, is involved in transcription of interferon. That's the classic pathway. But a new pathway came out, again from the James Chen lab, that was very intriguing to us. They argued that this process also stimulates autophagy, and they linked the autophagy to innate immunity. We confirm their data. If you see, if you look at, if you add cyclic GAMP, which will mimic this production right here to activate STING, if you simply add that to cultures, you can see an increase in LC3 lipidation. That's this lower band. And this is a signature of autophagy, but it's an equivocal signature. It's been shown in other processes. But this is the idea. This is causing autophagy. You take STING knockout cells, it doesn't occur. And if you rescue the STING knockout with GFP STING, you see increasing concentrations of cyclic GMP. You get a very robust lipidation of LC3. And then this is extending James Chen's work. We confirmed and extended that you can knock out most of the autophagy machinery and it doesn't affect this pathway. So this is a very strange pathway. The only pathway that's maintained in requirement is the ATG16L1, ATG5. These are the actual enzymes that cause the lipidation of LC3. And then the first break we had in finding out something new was when we looked at this baflomycin, which is an inhibitor of the F1 of the vacuolar ATPase. So baflomycin inhibits the ATPase on lysosomes and it prevents the low pH. People generally use it to prevent the degradation of this LC3 lipidation. So if we starve cells, which induces normal autophagy, we see that the lower band is basically eliminated. So people typically, this is a very standard tool to add baflomycin A to prevent the degradation. And here you see a shift from the top band to the lower band. That's normal. So we looked at this in this cyclic GAMP-induced process. And what you see is that if you add cyclic GAMP, you get the lower band. But if you add baflomycin, it doesn't increase like here. In fact, it blocks it. Very surprising. Now, if we add chloroquine, another way of blocking the pH, you see the lower band appears. But if we add chloroquine instead of baflomycin, we see the normal stabilization. So what is this doing so specifically? It's not the pH of the lysosome. So it's known the structure baflomycin binds the VATPase right here. We know it's localized to endosomes, lysosomes, secretory vesicles, and the Golgi. We looked at the localization of LC3. I showed you a Western blot, but here's LC3. If we add cyclic GAMP, we see it goes to these foci, and we see these foci nicely overlap with the VATPase. And if we add baflomycin, it prevents this focal localization. This localization also localizes with STING. So we thought we originally were puzzled, thought we were in new territory, but there's actually a couple examples in the literature of this called FIP200 independent baflomycin sensitive LC3 lipidation. And one is from the Flory group, Oliver Flory. He was a postdoc in New York, and now he has his own lab in Cambridge. And he found there's a VATPase-induced LC3 lipidation endosomal. And then he has a subsequent paper showing the WD40 domains required, and it occurs on single membranes. And this recent paper from Feng Xiao in China shows very similar activities, sensitive to baphlomycin A, and it's by the VATPase. And also it requires the WD40 domain. So I won't show the data, but for the STING-induced activity, the WD40 domain is not required. It's not required for starvation, but it is required for STING. So we're getting closer and closer intellectually to these prior papers. So we looked to see if it was going to double membrane autophagosomes or single membranes. And we use CLEM, correlative light EM. So these green spots are the LC3 puncta induced by cyclic G, A, and B. And if we do EM in that same spot, we can magnify it up very nicely, and we see it's these small vesicles. And here's even a further magnification. So the LC3 is going onto these small vesicles. And in the same experiment, the same cells at the same time, starvation induces autophagy in the same scale. You see the autophagosome is much bigger, and it has this canonical double membrane. So this lipidation appears to be not linked to autophagosomes. And this was done by my former postdoc, Mike Lazaro, and his new postdoc, Padman in Australia. And then one last point I think is really interesting is this paper I mentioned earlier that shows an analogous pathway. They discovered this based on a bacterial infection model, and they found that a normal virulence factor from bacteria called SOPF, they found this ADP ribosylates the VATPase, and it prevents it from recruiting the ATG16L1 and prevents it from this autophagy-related pathway. So we looked at SOPF, this bacterial-produced protein, and if we look at starvation, here's no starvation, here's starvation with baphlomycin, you see the band increases. Add SOPF, we get the same result. So there's no inhibition of autophagy by SOPF. But if we look at the cyclic GAMP-induced lipidation, it's completely blocked. And I won't go into this for the sake of time. But so this bacterial effector that blocks the ADP ribosylation, that ADP ribosylates the VATPase, prevents this STING pathway. So here's just I got a couple summary slides. So STING moves from the ER to these single membrane vesicles. And one mystery for the future is somehow STING activates this VATPase, completely unknown how. And it is involved in recruitment of this autophagy component, ATG16L1, that lipidates LC3, and this is prevented by this bacterial effector. So this is really mediating, sensing and mediating the LC3 lipidation. And a number of papers link this process to innate immunity, but it's not so clear how. And these are 3 papers that cover that topic. And then lastly, since I showed that loss of STING will rescue loss of Parkin, and I showed you that the SOPF pathway prevents one of the STING pathways, it suggests some really fun experiments going forward. So this is a summary slide. The STING has 2 routes of innate immunity. It can cause interferon production, or it can cause what we call VAL, this VATPase LC3 lipidation. It causes both of these. So which one of these is involved in exacerbating the loss of Parkin activity? So you don't have Parkin, you get phenotypes. If you knock out STING, it rescues. So STING is mediating some of these phenotypes. Which part of STING? The VAL part of STING or the interferon part? So we have a very nice tool now, because the SOPF factor that I mentioned right here, SOPF, it prevents this step, but not this step. So we're doing those experiments right now. And so we're going to tie this back into Parkinson's disease. It's going to be, I think, important tool. And with that, I'd like to thank you and thank my group. I mean, I've had a long list of former postdocs and graduate students that have unraveled this pathway that I don't have time to discuss. This is my current lab. I just want to shout out to Andy Molman, who did the unpublished Drosophila work with Sting and Parkin, and to Tara Fisher, who did this Sting LC3 lipidation veil work that just came out online. And also I'd like to acknowledge Jobert Vargas. He is a graduate student of mine in the joint UCL NIH program. He's just finishing up next week. I'll be listening to his Viva online. He did his work with Jippy Shavo. And with that, thank you for your attention.
So I'd like to thank you, Richard, for a fantastic talk and for your fantastic work, and congratulations on winning the Breakthrough Prize. I'd like to now ask the President and Provost, Michael Arthur, to give a vote of thanks. Thank you.
Mike, thank you very much. Before I turn to our guest lecturer to give a vote of thanks, can I just say, Mike, that was an amazing list of achievements for the Institute of Neurology for the past year. And I just wanted to pass on my congratulations to everyone and my thanks for such superb and excellent performance in the face of adversity. And then also thank you for the very kind words about me and my departure. I'm very grateful for that. I wish the Institute of Neurology all the best for the future. I will be, I hope, involved in helping you open that building when it's all done. So it's a farewell from me this evening rather than a goodbye. However, my main role tonight is to thank our speaker, Richard Ewell. And Richard, it's tough giving a lecture online like this. We would much rather have had you in the lecture theatre at the Institute of Neurology so we could get to meet you, shake your hand, and become your friend. But I have to say that you didn't flinch from using the technology and illustrating your lecture absolutely beautifully. So thank you very much for making that effort and for giving us a superb lecture this evening. Before we started with the lecture, Richard and I were talking about the Breakthrough Prize. And of course, we know a little bit about it because of our own Professor John Hardy being a winner in the past, and that's led to a number of us being invited. And I was asking Richard what was happening to the big party and the Oscar-style ceremony. And of course, because of COVID, that's all been put off for a year. But Richard, I can tell you when you get there, it's a wonderful event, and you should revel in the occasion because it is a prize that's very difficult to win, and it involves a lot about as my understanding anyway, that it's about a sustained contribution to understanding an extraordinarily complex and difficult problem. And to the relative lay individual listening to your lecture, that was really, really evident. And I was going to try and, summarize my knowledge of the pathway after you went through it, but there were so many molecules and so many links and jumps, I couldn't quite make it. However, when I turned to the Breakthrough Prize website for some simple form of words to summarize what you've achieved, it says that you've achieved the Breakthrough Prize for elucidating a quality control pathway that clears damaged mitochondria and thereby protects against Parkinson's disease. And the way in which you skipped along that pathway with those beautiful experiments was just lovely. The one that impressed me as a relatively lay individual in this area was the slide of the mitochondria, and then as you depolarize, eventually the mitochondria just completely disappearing. That to me was just so totally convincing of the importance of this pathway. So thank you very much. We're very grateful to you for joining a very distinguished list of previous guest lecturers for this particular occasion. Your lecture was just absolutely superb, and we're very grateful to you. And thank you very much for making the effort and for talking to us this evening. Thank you.
So thank you, Provost. So it just is for me to also add my thanks, and I'm sure the thanks of the entire audience. I know there was around 300 attendees. To thank you, Richard, for a fantastic talk. So that really concludes the annual address for this year. Thank you for attending it. And it just remains for me and all the Institute management team, to wish you all a very happy Christmas and hopefully a better new year than this last year. So thanks for attending and happy Christmas. And that's the end of this annual address. Thanks a lot. Bye.